Today is July 23rd, 2014. I'm Kevin Foskett, the chair of the Department of Physiology at the Perelman School of Medicine at the University of Pennsylvania. And today I have the distinct pleasure and honor of interviewing Dr. Robert Forster for the American Physiological Society's Living History Project. Robert has been a member of the APS since 1946, nearly 60 years ago. He was the 39th president of the society and was the chair of my department, this department, the Department of Physiology, from 1959 until 1990. Robert received his medical degree in 1943 from the University of Pennsylvania and went on to become one of the leading respiratory physiologists of his time. In this interview, you'll hear about the development of his career as well as his research program and the other individuals with whom he interacted during that career. Robert, welcome to the Living History Project, and thank you very much for agreeing to be interviewed for this series.
A great honor to be here.
Robert, would you mind sharing some information about your early life and those influences, perhaps books or your parents that contributed to your career development?
Well, first I want to give a caveat that I have a poor memory and I need notes, and the dates are maybe inaccurate. I had a very successful dentist and civic worker who taught me facts and encouraged me to disgorge them and show them off at family dinners. I looked like to make things. I cast lead soldiers. I took apart a wristwatch that was a present and could not reassemble it, so I guiltily hid the corpse in my sandbox. I would make solid working Canon models, which I sold to my classmates at school. I was later an amateur radio operator, W3FFQ, and I actually built a large transmitter for my high school. I got into science fiction. My science teacher had a trove of them I discovered in a closet and started my interest in science fiction, which has always been here. I had good friends. This was not quite a nerd, but my parents were always supportive, very supportive, but the science was not exactly their style.
You received your Bachelor of Science degree from Yale University in 1941 and then your MD from the University of Pennsylvania in 1943. So those were the war years. Can you tell us how the war influenced the timing of your degrees and what impact it had on your research?
Well, I wanted to hurry, and I got out of college in 3 years in 1940 before World War II started. The first year of medical school was substituted for the last year of college. I went to Penn Medical School because I could live at home and save money, which I did until the Army took over. Physics interested me. My goal was then to be a radiologist, not for the money, which I understand today is quite good, but for the physics. World War II really affected me first by paying for half my medical school education in the Army student training program. We were put on in uniform and had a semblance of Army life, but my war was actually spent in a laboratory.
So as a medical student, you had some interest in research. Can you tell us about working with Julius Comro and Carl Schmidt during that period of your career?
Well, I was supported by both of them very much. And Comro was a great teacher in pharmacology. I tried a little experimental work to investigate the effect of thiocyanate on blood pressure, which is rather a popular therapy at the time. And I assumed it was a change in plasma viscosity, which is absolutely asinine. I spent some time, got nowhere, but wrote a review of thiocyanate's effects. That was my first paper. During several summers, I worked in a laboratory at the Alfred I. DuPont Institute in Wilmington under Douglas McFadgen, a biochemist, and Marianne Javine, a pathologist, who were from the Rockefeller. I learned a little biochemistry and how to operate a Van Slyke apparatus, which was a bit of a help later.
You did a short internship at the Peter Bent Brigham Hospital, and then you entered the Army, as I understand it, and you were assigned to the Quartermaster Corps Climatic Research Laboratory. Can you tell us a little bit about the work that you did while you were in that laboratory, and maybe about some of the people that you interacted with there?
Well, George Thorne was my chief at the Brigham, and he had taken care of Howard Hughes, who had adrenal corticosinsufficiency, and was instrumental in founding the Hughes Foundation, and he recommended me to replace Cliff Barger at the Quartermaster Corps Climatic Research Laboratory, who had contracted TB. Our mission was to evaluate the effectiveness of soldiers' equipment, either by having soldiers test it in the field or measuring the temperature conductivity. And thereupon, my field became temperature regulation. The CRL was the brainchild of a General Dorio of the Harvard Business School and commanded by Colonel John H. Talbot, who was previously at the Massachusetts General Hospital, father and chief of a cancer hospital in Buffalo, and finally the editor of the JAMA. Dr. Talbot had physiological experience. He had been on a high-altitude exposition in Peru and was possibly a member of the APS. The CRL still exists in Natick, Massachusetts, to this day. I constructed, while I was there, a finger impedance plethysmograph and volume plethysmograph to compare the electrical impedance changes with the finger volume. That showed that the pulse records looked alike, but measuring the impedance of the finger was not a precise member of the volume. Its proponent was John Nemore, who was not at all happy when I came out with this. I never published it. Alan Burton was also a visitor, and he made scurrilous remarks about the impedance plethysmograph, to which Cuthbert Bazette said, "Pay no, never mind. I taught Alan all the physiology he ever knew." There was a Richard Day, who was a pediatrician and later chief at Columbia, developed a plethysmocalorimeter to measure simultaneously the leg's blood flow and heat exchange. In a cold room, Dr. Talbot and Cuthbert Bazette stuck a thermocouple needle in my radial artery. They didn't ask my permission, by the way, and showed the blood was cool before it reached the hand. This work resulted in a paper on page 3 of the first issue of the JAP, in which I was the last author. I have assumed, awaiting contradictions, that this was the first demonstration of pre-cooling in an artery. My World War II experience was just a step up in my career, by fortune, while my two brothers were fighting a dirty war, and my father was in the Air Force in Texas, for which I will always feel guilty. We also had Sir Hubert Wilkins, the Arctic explorer who went under the ice in a submarine, visit us.
Well, those sound like very exciting times. After the war, you returned to the Brigham as a resident and then joined the Department of Physiology at Harvard Medical School on a Life Insurance Medical Research Fund fellowship. At that time, you worked with Eugene Landis. Can you tell us about the research you did at Harvard?
I might add that after World War II, I spent a year as a graduate student in mathematics at Harvard, supported by the GI Bill. I learned a bit of practical math and physical biochemistry, but was devastatingly put down by a bunch of kids. I then had a residency at the Brigham, during which, with Louis Dexter, we measured the pulmonary artery temperature with a sensitive catheter, then a new technique, without asking anybody's permission, including that of the patient. It was a body temperature, of course, was rather useless. Knowing Glenn Millikan, who had been teaching pharmacology at Penn, I obtained one of his ear oximeters, which was the antecedent of the finger oximeter you see today, and measured the oxygenation of some of Dr. Samuel Levine's patients. Next, I went to the Department of Physiology as a life insurance medical research fellow under Eugene Landis. Landis got his MD and PhD in physiology at Penn. His name was spoken with bated breath when I was a student there. He had taken the micropipette technique of Newton Richards and proven experimentally the Donan equilibrium was correct. In the lab was Karl Gottschalk, who was using the same technique to measure osmotic pressures in the kidney. Dr. Landis wanted Benson Rowe and myself to first use a thermostomer to measure GI blood flow. After several unsuccessful tries, I thought that it wouldn't work theoretically, which did not please Dr. Landis. But I was allowed to carry on temperature regulation studies and elected to measure chronically a temperature regulation in the hypothalamus of the unanesthetized cat, where a temperature regulatory center is assumed to be. My approach was very physical. I assumed the hypothalamus acted as a thermostat. I constructed a thermistor at the end of a double-barrel glass tube, which I'd made myself, which fitted on a stainless steel screw in the cat's skull. The cat did not seem to mind this. The experimental setup was excellent. The results were iffy, but I got it published. I had obtained fine Constantan wire for some thermocouples. Thermocouples are copper and Constantan. When I did not realize it had to be specially drawn, and it used up all the extra funds on my scholarship fund, which certainly did not interest Dr. Landis, and I received a dressing gown. I still have the roll of wire as a reminder.
So after your fellowship, you looked for academic positions, and fortunately for us, you were hired in the Department of Physiology under Julius Comroe here at Penn. Can you describe the years, those years in the department, and talk about some of the people that you worked with and maybe others in the department at the time?
I had sought a post in several departments of medicine without much success. At Johns Hopkins, my interviewer stopped in the middle to say he thought I was applying for medical school, which did not discourage me. I eventually got to see the professor who offered me a residency, since I just had a residency and been through the war. I was disappointed. Then suddenly, out of the blue, I received an offer in physiology in the Graduate School of Medicine and anesthesiology. I was then in surgery from Julius Comroe and DRIPS combined, with a handsome salary. The salary was so handsome that Dr. Landis thought it was indecent. Of course, I accepted. I gave up the mechanical theories of temperature regulation and overnight became a pulmonary physiologist. Julius Comro and Seymour Kenny had great foresight. They had taken advantage of new instrumentation that occurred during the war and had ordered and purchased an intact infrared CO meter and a breath analysis mass spectrometer with which I was to repeat Marie Krog's use of CO in measuring lung diffusing capacity. They also had a magnificent steel plethysmograph built, which Arthur Du Bois took on and got to measure many mechanical aspects of the lung which are being used today. The lab was at the peak of its fame as a center of lung physiology and testing, attracting physiologists and clinicians from all over the world. But first I spent several months tinkering with a mass spectrometer, seeing if I could increase its sensitivity, until finally Julius sent in Ward Fowler to see if I could do something useful. I had spent several months learning respiratory physiology with John Severinghouse, Ward Fowler, and David Bates. I made no physiological measurements, but when Ward came in, he suggested we use the dilution of an inert gas to calculate the initial CO and blow out a sample for the second sample of CO. The changes in CO concentration in the breath were so great that the errors that I was worried about were not significant. With this, the single breath diffusing capacity was born. We made a series of studies on CoExchange, in which I had some equations. Colin Ogilvie had finally standardized the single breath method and showed it was clinically useful. Then Norman Staub and I went back to oxygen and studied its uptake and distribution in the lung. Masaji Mokuzuki came as a fellow from Japan and worked with me on CO uptake and hemoglobin films. This was the start of a wonderful series of Japanese workers who came to study with us. And the last one was Yoshiaka Uno. These friends invited me to Japan for several times. While this was going on, Julius was working on a simple presentation of clinical pulmonary physiology with clear illustrations, and Arthur Du Bois, Bill Briscoe, and I prepared separate sections. The result was the lung, a bestseller, and gave fame to the department and all of us. It went to 3 editions, the last with Aaron Fisher. We were fortunate to have available pressure chambers operated by Chris Lambertson, the predecessor of which had been used to study high-altitude physiology on my medical classmates by Julius and Carl Schmidt. Dick Hyde, Chris Lambertson, Gordon Power, and I measured pulmonary diffusing capacity and blood flows at pressures up to 3,200 millimeters of mercury. I also pored over several papers on CoExchange by Professor Francis John Wordsley Roughton from Cambridge, particularly his estimate of the transit time of blood in pulmonary capillaries from measurements of CO uptake. His argument was convoluted, and I managed to reduce it to an equation, but needed more data on the rapidity of CO uptake by red blood cells, for which I wrote him. The upshot was his coming to our department, bringing a new instrument to measure the rate of CO and oxygen uptake by red cells via the Air Transport Command, which paid no attention to the admission to either country. His new instrument was rendered useless by light scattering of the red cells, and Bill Briscoe and I built a continuous flow rapid reaction analyzer with suggestions and a beam splitter from Britton Chance. The net result was 4 papers in the JAP, which separated the pulmonary blood volume and diffusing capacity of the membrane by measuring the diffusing capacity with CO at different oxygen tensions. All of this, I might say, resulted from the instruments which had been visioned and obtained by Comro and Ketty before we ever came, and the know-how of Roughton. And this was not from any great brilliance on my part. Incidentally, Ronald Coburn, Peter Kane, and I developed to calculate the exchange of CO in the whole body, which is very much used today, and has the unusual name of the PFK equation. This propelled my research into the rate of gas exchange with red cells, not only CO and oxygen, but later nitric oxide. Measurement of the rate exchange of CO2 with the red cells, of course, led me into carbonic anhydrase, which Roughton had discovered. We could not determine the correct rate inside the cell because of a limitation of flow across the membrane, both gas and bicarbonate. But again, fortunately, Tomo Itada, who had worked with Mildred Cohn on a mass spectrometer, came into the department as an assistant professor, and I was able to purchase Mildred's discarded mass spectrometer from Britton Chance, who didn't own it, in fact. Tomo taught me about mass spectrometry, and we measured CO reaction rate in solution by 18 oxygen exchange, which is the method that Urey had developed at Chicago. Eventually, I derived an approximate equation to measure the correct reaction velocity in the interior of the red cell, and thus we were able to measure the catalytic value of Ca that was in the cell. And plus, we got as a bonus the transport of CO2 across the cell wall. With Lena Mila, having just learned to isolate mitochondria, Susanna Dodson, Baird Story, and we thought to test out the ideas just by coincidence, and found carbonic anhydrase in the mitochondria. Gerolf Gross was invited as a fellow from Germany. On the stop flow pH instrument, we measured the rate of CO2 combination with hemoglobin, a reaction that Roughton had proposed, which many doubted. He returned to Germany, became the chairman of physiology at Hannover, developed a simpler mass spectrometer, and has applied the 18 oxygen method widely, extending it to estimate the permeability of the membrane to CO2. I said above it was the permeability of bicarbonate we were interested in. Susanna Dodson was recommended as a fellow by a collaborator in Australia and applied the 18 oxygen method using a larger and fancier program mass spectrometer to biochemical studies, teaching me biochemistry in the process. I was very averse to biochemistry in medical school. She became a great protagonist of carbonic anhydrase, which had been studied by many molecular biological techniques and shown to be very ancient in all life, in all tissues, and had a myriad isozymes. With this spectrometer, we could continuously monitor changes in isotopically labeled compounds, which led us further into biochemistry, which Yoshiaka Ono has carried much further. I was diverted into the field of CO metabolism by a surgical friend named Bill Blakemore. He had been in Sweden on a fellowship and brought back the very radical idea of Sjöström that carbon monoxide was actually made in the body. He had some equipment with him from Sweden. Ronald and I distrusted this method, and I proposed a very complicated theoretical method to measure CO production. But Ronald proposed a very simple one by having the patient breathe in or individual breathe in a closed system and measuring any increase in blood carboxyhemoglobin. His method found an increase in body CO of about 0.4 of a milliliter an hour, and eventually, and it is used today. He then showed that the CO came from the degradation of a heme ring and got the enzyme that would do this. Not the enzyme, but the site from which it came. Ronald Estabrook and the Johnson Foundation then discovered that the cytochrome P450 existed in the liver. He had discovered the liver enzyme, which is a band at 450, and this is known as the P450 enzyme. He and Ronald and Maria Della Varia showed that many rings and hormones are degraded, producing CO, which provides a handle for their study when many drugs are detoxified the same way. And Larry and Longo and Maria went on to study placental exchange, some of it at high, elevated PO2 pressure in the chambers. I think that ends that.
So your chair, Julius Comro, left in 1957 to establish the famous Cardiovascular Research Institute at the University of California, San Francisco. And then you became the chair of physiology in the Graduate School of Medicine here at the University of Pennsylvania. But at the same time, John Brobeck was the chair of the Department of Physiology in the School of Medicine. So were there any conflicts in having two departments of physiology at the University of Pennsylvania?
Well, I'll deviate a little bit to say Julius was frustrated in his plan to expand the Graduate School of Medicine and left for San Francisco in 1957, and George Coley became chairman and shortly made me professor. 10 years later, he was appointed chair of pharmacology in the School of Medicine, took pharmacology in the graduate school with him, and made me created the Isaac Ott Professorship out of some funds which are available to him as Dean of the Graduate School of Medicine, and he could do that at the time. The two physiology departments operated in remarkable harmony during their coexistence because John was a very lovely fellow. That in the School of Medicine was in neurosciences, cardiovascular, GI, and muscle, while the Graduate School of Medicine was still largely in pulmonary.
In 1970, Brobeck stepped down and the two departments were combined under your leadership. So can you tell us about that period of time and the people who were the faculty in the department, in the combined department?
The agent really was Dean Alfred Gellhorn of the School of Medicine. He was Ernest Hemingway's brother-in-law, which he was never proud of. And the great interest and cooperation of John Brobeck, and he combined the physiology departments and made me chairman. I did nothing, but was benefited really by John Brobeck, Alfred Gellhorn, and George Coley. John told me once that he did not learn why Julius had been passed over for physiology in the School of Medicine until after he came to Penn. and would not have come had he known it, and I guess he always felt guilty about taking the position. The muscle interests of the School of Medicine department were expanded by Sol Weingrad and Anne-Lee Somlio into a muscle institute, led later by L. Goldman. Frank Brooks continued his GI research. The neuroscience interests of John Brobeck, Grayson McCooch, unfortunately, were taken over into the new Department of Neuroscience. The new area of membrane physiology was sparked by Ernest Kleinseller, Mortimer Savan, and Clay Armstrong, and Lyle Peterson carried on the cardiovascular interests at the Graduate Hospital.
You were chair until 1990, when you stepped down. So can you comment on your life since then?
After retiring, my successors have treated me very kindly. I kept my office and laboratories and actually still have them. My funding continued for a while, and I continued work mainly related to carbonic anhydrase with Gerald Gross, Susanna Dodgson, Sukumi Luhiri, Robin Post, and Baird Story. Yoshiaka Ono, Taguchi, Shoko Nioka, and Anjan Zimmerman were all very, very helpful. Krista Vanderberg and Christoph Roblewski, who are not here now, she's in Florida, introduced me to the use of magnetic resonance spectroscopy of 31 phosphorus. And they, with Post, Lahiri, and I studied the metabolic action of carbonic anhydrase in living muscle. Later interest in nitric oxide arose because of its extremely rapid reaction with hemoglobin. The idea that its uptake in the lung could depend only on the diffusion through the membrane and not on any reaction in the blood. Botras and we measured the nitric oxide uptake in red cells, however, finding only a little bit greater than that of oxygen, which he presented at FASEB. Unfortunately, these data were too variable to publish. I was going to get more precise data, but someone broke into my lab last summer in a part of the action against toxic compounds, which they considered liquid mercury falsely was. They broke some of my equipment and was destroyed, and that was the end of my career. Francis Tay Nuyenhouse had come from Holland, and we went back to carbon monoxide and continuous flow rapid reaction apparatus and measured the uptake of CO in pig blood.
You mentioned earlier that you've been associated with the APS for many years. In fact, your first APS meeting was in 1946 in Atlantic City, New Jersey. Can you tell us about that meeting and how it compared with meetings that you've attended subsequently?
I was elected to council in 1963, and Herman Rahn, John Pappenheimer, and John Broeck were with it on me. At that time, one of the council, possibly the president, paid for the group dinner. You compare this with the tremendous bash the president throws today. Now, we thrashed out a problem of the APS representative to the International Union of Physiological Sciences. It seemed that a president of the APS had recently gone to an IUPS meeting abroad and was frozen out of the official meetings because APS had sent a representative to the IPS who hung on to the post. In spite of a large meeting support for the meeting by the APS. The president, of course, was irritated. We changed our relation with the IUPS. Years later, I wrote the member who had been the APS representative to IPS and thanked him for his efforts. He was very bitter at the time, thanked me and said it was the first word of gratitude he'd ever received from the APS. I also remember a wrangle when Wallace Fenn was trying to buy the land on which next to Pook's Hill, on which the APS and the federations exist. The bulk of the APS nest egg he wanted to use was in the publications fund, and the publication board were very uncooperative. By some parliamentary legerdemain involving Alfred Fishman saying he had changed his vote, the APS got control of the publication board's funds and purchased the lands where you all sit. About 1963, Morris Vischer fulfilled his dream of organizing a series of APS handbooks, and I wrote several articles for the respiratory section, which was edited by Fenn and Ron.
So you sat on the APS Council in 1963, and then you served as the Society's 39th president in 1966 to 67. And you've told us a little bit about the Society during that time. Subsequently, the APS hosted the 24th IUPS Congress in Washington, D.C. in 1968, and you played a critical role in having the Congress since the IUPS president, Wallace Fenn, asked you to be chair of the Finance Committee. Can you tell us about the Congress and the participants at that meeting?
Well, it's said that I was very significant, but the facts are I drifted along. Wallace honored me by asking me to chair the Finance Committee, but he had the foresight to include K.K. Chen, the pharmacologist, who had, with Carl Schmidt, I believe, introduced ephedrine in the country. And he knew somebody in every pharmacological group in the world, and money just poured in, and all I had to do was sign checks. My wife and I had the greatest time we ever had. We had a magnificent hotel suite with luxurious bedrooms for the Pappenheimers and the Forsters, with a large couple of living rooms in between, in which a continuous series of cocktail parties for Physiology's great from all over the world were had. Wine and hors d'oeuvres were liberal. Of course, there were many scientific presentations, and the meeting was a great success.
Sounds like it. Well, you've had a great career scientifically and professionally. In reflecting back on it, what would you consider some of the most interesting or surprising findings in your research?
Well, I would say that the most significant things, I think, were regarding CO and the measurements of human clinical conditions, and also that measured the volume of the lung capillaries, which I believe is the only method for measuring capillary blood volume at any time, and that the resistance to much of the uptake of oxygen and CO was in the red cells and not in the membrane. Also the ability to measure with 18 oxygen the CO2 reactions inside the red cell, in spite of the membrane impediment, was very useful. And CO production in the body, which has led to a large series of studies on the many, many things that break down the heme in the liver. And finally, the rapid uptake of nitric oxide by blood, the fact that it is not much greater than oxygen in spite of its tremendous reaction velocity constant.
What role do you feel that the American Physiological Society and perhaps other professional societies that you belong to have played in your career?
Well, I say the APS is my society, and I've been active in it and met some wonderful people, including Ray Daggs, for almost 58 years. Most of my papers have been its publications, and its meetings kept me up to date. And I am simply amazed at its great size and wealth today. The Society for Clinical Investigation spanned between science and medicine for a while. I published some there, but I was less comfortable when I left the clinic. I think the APS should be careful to keep its finger in organ and system physiology, of which it arose. That is what makes us unique and essential to medicine. Physiology has given rise to biochemistry, pharmacology, and neuroscience, and these sister disciplines are all today in molecular biology because of the new excitement and the money, and it's hard to tell them all apart.
During your career, the environment for doing research has changed many times, and I wonder if, looking at at your own history, whether you might have any advice for students considering a scientific career today.
Well, I would think, particularly in view of the funding problems, I would not go into research unless you love it. Too many are doing pedestrian research to make their way up the academic ladder and then end up in administration. Your teachers are your foundation. You only add a little to what they taught you. And luck is most important. If you look back over my career, I've had extraordinary good fortune, and a great many people helped me. But you have to recognize it when it comes. Keep your eye open for something new. If it's really new, nobody else will believe it, and all the common thought is against it. Just remember finally that a new finding gets incorporated in the body of science because it becomes an accepted fact. The excitement of its discovery disappears. You will be forgotten, but hopefully your findings run on.
Robert, thank you very much for your willingness to be interviewed for the Society's Living History Project. It's been an honor. It's been fun to learn about your life and your career and your ideas about science and research. Thank you again on behalf of the Society.